I think it's time right now. It's 11 o'clock. So good morning, good afternoon, and good evening, everyone, no matter where you are. Welcome to our NovoMagic webinar, Introduction to our AI-powered bioinformatics assistant. I am Carolina Jan. I am Senior Product Marketing Manager at Novogene Europe, and I'll be your host today. And with me, there's our lovely colleague, Dr. Wenjie B, who is our bioinformatics consultant also at Novogene Europe. Now, while we are trying to wait for all the attendants to join our webinar, I'll just give you a really brief introduction about those who don't know about Novogene. So here is an overview of today's webinar. I will just give you the short introduction, move on to the service workflow, and then I'll just hand off the mic to our speaker today. Now, Novogene, as you know, it's a global leader in NGS services. We have developed 10 laboratories around the world. We also have 90 subsidiaries with offices and local support teams for you. And we are proud to say that our business covers around 90 countries across 6 different continents. Now, Novogene Europe, we are based, of course, in the Europe continent, and we have 2 labs. We have one in our Cambridge Sequencing Center, and the other one in Munich, which is part of our Novogene Germany subsidiary. Now, we are also very proud to say that we have worked with 100% of the world's top 100 research institutes, and we are now also working with top 10 global pharmaceutical companies. And we're also very happy to help with small and medium-sized companies to improve and thrive their research results with our NGS services. Now, we work with the latest sequencing platforms. We are officially certified service providers for Illumina, PacBio, and Oxford Nanopore. And soon we might also be incorporating new emerging players. So please stay tuned to our news. And here is just a brief look of our Novogene Europe sequencing services. And as you know, you can find more information on our website. But recently, we have had some new additions like all-link proteomics, spatial transcriptomics, single-cell RNA sequencing, and enzymatic methylation sequencing. Now, just before I pass on the mic, I'll just give you a really short introduction to our service workflow. And as you know, we offer end-to-end service workflow. So whenever you have a project inquiry, feel free to reach out to us. And from that point, our local representative and our technical support team will be in touch with you, and we will guide you through the entire process. So from the very beginning, from sample information form, how to ship the samples, to the very end, the data delivery process, we will be tagging along with you. And even if the project ends, we will also have a large window for after-sales issues, so you will feel supported during the entire journey. Now, regarding our messenger RNA sequencing service workflow, which is specifically explained for this webinar, we just want to showcase that we have this experimental design where we can help you address these several questions. What kind of goal you want to reach? Do you want to quantify the genes? Do you want to identify the function of those transcripts? And we can also help you evaluate extraction services and assess you with the correct library preparation method, the correct data amount, and of course, the bioinformatic pipeline that you need. So that would be all from my side. Wenjie, whenever you want.
Hi, everyone. Greetings from my side. So that I'm a bioinformatics consultant in Novogene, and today I'm going to introduce you the standard bioinformatics pipeline in Novogene and also the NovoMagic platform, what you can use for the reanalysis of your data. First, the introduction from what do you have after the sequencing. You got the raw reads. It's need to go tons of analysis until you can interpret the biological insight in there. First, we will do the quality control to control the error rate, GC content, and filter the out of the contaminated data. Then use only the clean reads for the mapping to map to the reference genome. After using the mapped sequence reads, we could quantify all the genes. And using quantified data, we could have an overview of the quantification to see how all the samples are correlated with each other, and in the PCA plot, see how they are separated, how they are differentiated from each other. And then based on the quantification data, we can also do differential expression analysis and use this data, then we can continue with the functional analysis. DO on this GNet, as well as oncogene analysis and protein-protein interaction. In addition, we also offer the structural analysis like ternary splicing, SMP indels, and fusion gene analysis. I would say this analysis can be distinguished into two different parts. The first part is the basic analysis, what the majority of the RNA-seq would need it already. So the quality control to remove the low quality reads and adapt the contamination, and then to do the mapping to align the reads to the reference genome, and then quantify the reads to count how many reads align to each gene or transcripts, then continue the downstream analysis to identify differential expressed genes and their biological meaning. And then there will be advanced analysis. These are only to apply to the part of the project. You only need it if you're specifically working on this, like protein-protein interaction, to analyze functional relationship between the identified or quantified genes and proteins, and oncogene analysis if you're working on clinical sample, or mouse models working on to identify cancer-related genes by referencing certain databases. And the alternative splicing, if you're interested in working to detect different exon usage patterns and the genetic variation to identify the single nucleotide polymorphisms and the small insertions and deletions, as well as the fusion genes, and which will also needed for clinical samples or mouse models if you're working on cancer. And the first step would be the quality control. What you see in our report we offered you would be these three plots: the data filtering to make sure that the majority of the reads are clean reads, so low contamination, low adapter contamination, and the majority of the high quality reads. And the error rate will be also controlled. As you see in the position along the reads, the error rate is increasing because the limitation of the sequencing strategy itself, but it's always under control, always lower than 0.05%, and showing the good sequencing data. And the ATGC content to see that all these bases were stable over the all the sequencing except a few bases at the beginning, which is quite normal. And these signs are indicating the good sequencing quality, so we can continue with the clean reads downstream to align them to the database. Here for the alignment we use the HitSet tool. He said using the strategy that to first do a fast round of scanning to discover the splice site, and then the second round of, we use this splice size to make all the reads aligned. And using this alignment reads, you can get the raw quantification abundances, but to visualize them across the samples, we need to normalize them. Here we're using FPKM, so fragments per kilobase per million. It is adjusted based on the reads of the length of the genes and also the depth of the sequencing, so that you see that across all the samples, the box plots distributed are quite similar. You know that the sequencing went quite well, and there were no samples had obviously bad quality. And based on this expression quantification, we could check the Pearson correlation among other samples. So the closer the value to 1, the higher, the more similar samples are to each other. The lower the value, and the more distinct they are from each other. Here you could check the controls within each group. For example, like the controls, they are quite similar to each other. They have very high value, so that are quite similar, which is a good sign that the samples are belong to the group, are clustered together. And another aspect is to look at the PCA analysis, principal component analysis, to see how the sample is distributed. The PCA is just a way to pick through your data to see how the most variance are distributed. So for example, PC1 has a 64% of the variation explained, PC2 has a 17% explained, and the controls are far away from the other samples, which is a good sign. And that is, and then we can continue to compare the different groups using this, for example, generated this volcano plots. Volcano plots has access as log2 fold change, and y-axis is minus log 10 p, just the value. So that it's named the volcano plot because the data points are like erupting, so that the most interesting genes of at the upper right and upper left corner, which has a higher fold change and more significant regulated. The software we use for biological replicates, we use DSeq2. If we don't have biological replicates, we do use HR for it. And putting all the up and down regulated genes, we could generate this heat map. So you can have a better overview of what's happening here across all the samples. The red of the color, the high expression, the green, the color indicates low expression. And you could also give us the metadata, the other information, we could map them together. So you see what's exactly happening in your samples. And in this map, each row would indicate the gene. And here you could see that we have thousands of genes. To interpret the biological meaning, we need further analysis, meaning the function analysis to see, for a set of functional genes, how they enriched in this significantly regulated list. For example, you can imagine them like you have all the background genes set there. A certain of them belongs to this pathway. You have this up and down regulated genes set, and how many of them belong to this pathway? Then, and if it's just happen by chance, or is much higher than that. So we get a p-value out of it. So each function, each pathway. And then we could create a plot like this, scatter plot with x-axis, the gene ratio, y-axis for the each function, each pathway, and then you could see which function, which pathway are mostly enriched. The size indicates the count, and the color indicates the p-value. You can get most of the information to see what's happening in the samples. There are also other plots available, can help you to interpret data like bar plot, and but here's just example. You can see all the information later. And if you're working on alternative slicing, we also offer the analysis. Here's an example that the skipped exons, there's 3 exons available here. There's some reads would include the exons, having this as inclusion as a form. Some other reason might skip the middle one and have the skipping as a form. And in the end, we provide you the plot like this to interpret the data so that I have these 2 isoforms, 3 exons. This middle one could be skipped. So that you see in this sample that the majority of the reads have the connection among these exons, while in the other samples, there are less connection with this middle. So in the end, we have the inclusion level for the sample higher. That means the is more likely to include this exon in the middle, and the other sample has a lower inclusion level, and it's more likely to escape this exon. Another service we're knowing here is to find out the genetic variations, for example, the SNP and indels, which are also valid identification using the RNA-seq data. And we offer you to classify them. For example, the regions where they happened, exons, introns, or intergenes, and what are the functions? Are they methyl-senses, or the non-senses, or just the silent mutations, or the potential impact of them for the protein function? And the fusion genes, if you're working on clinical samples or mouse models, work on the cancers, they might be interested for you, because fusion genes happened in the chromosome rearrangement and could induce a cancer. We quantified the fusion genes happening using the span reads, which for paired reads that spans at both loci of the genes, or using the junction reads, so that a single read could span this junction site. Based on these reads, we could create this Soros diagram for you, so that outside is all these chromosomes available, and inside we show you where the junction site is located. Then the blue line indicate the fusion genes happens between chromosomes, and the green arc indicates the fusion gene happens inside each chromosome. And although we provide you all this professional analysis, happens quite often. You need to reanalyze the data, and when and why? Because it happens very often you label the samples wrong, for groups them wrong, and maybe you find some poor quality samples in the PCO heat map there, and you want to remove them. And maybe the boss asks you to try a different analysis method. Maybe you got suggestions, maybe there's a new tool coming out, maybe you want to try it. And if you find a specific pathway of genes that are much meaningful, you might want to rerun the enrichment with a new gene set. And in the end, we got all the data, you might want to improve the figure layout for publication. You might want to adjust all the size, colors, or scales of the figure. And of course, you can do themselves if you know how to coding, or you can ask a bioinformatician. But if you have a deadline coming next week, then it's difficult. So reanalysis is often unavoidable. And the key is how fast and how flexible you can do it. And if you're our customer, you have these options to send to our bioinformatics team, so that you can request the removal of some bad samples, regroup them, and rerun the complete analysis. It's perfect fun, and this data will be handled by our expert, but might take some time, days, or sometimes even weeks. But most will cost you money if it's not our fault. So that here we could offer you another magic to you, so that NovoMagic platform would fulfill all the wishes as above, so that redefine the groups, remove samples, and use a different tool to rerun the enrichment, adjust the figures with no delay, no extra costs, and you don't need any coding knowledge. You can take the full control. We have the AI powered regent, so that you can just dictate and make the changes, and the output will be flexible for your presentation. And here we introduce you the NovoMagic Plus. But if you're already a customer like years ago, you might know the NovoMagic legacy version, which is also cloud-based NGS data analysis and visualization, and it's already offered you some certain functions, and a lot of customers liked it, like the analysis I mentioned above, they are almost supported to do the gradation analysis, clustering, to visualize the data, to show you the volcano plot, the heat map, and so on. They were quite helpful, but there are also certain limitations, like these pipelines were almost all fixed, and there are limited figure editing you can do there. You cannot change the layout, and the transcript, it was only applicable to the transcriptomics, and the layout was traditional and form-based. There was no AI available. From this year and from April, we have this new NovoMagic Plus coming out, which support you the flexible grouping to adjust all the parameters, any parameters you like, and create a customer report. And it is fully support the customizable reports. You can change any detailed parameters in there until you're satisfied. Currently, it's also support only transcriptomics, but amplicons and other toolkit modules are coming soon, probably at the end of this year. And it has new platform has modern, cleaner layout and has an intuitive interface, which I will show you later. But the most important difference is the built-in AI assistant Novi here, which I will immediately show you what it does. So Novi is developed by Novogene and is supposed by Bioinformatics to visualize our data and to generate the plot, the transcript between the languages, and to give you the literature summaries. And so the motto is to make bioinformatics analysis more convenient for you. Novi itself, its core engine is powered by R, but it's more than just the R coding itself. Is supported by natural language, so that based on the raw data, it will give you the output directly. And this user-friendly design, you just dictate to make like the chat. Maybe you already use the other AI like ChatGPT. You're familiar with it. You can just chat, and it will generate a chart for you. And it's fully customizable. It gave all the details. And currently, we offer these modules for the Novi: Bioplot, TechTalk, image to code, and paper polish. And you can guess the function already from the name. So the Bioplot will generate a ready-to-use figure for you, and TechTalk will answer you all the technical questions or any scientific questions. And image to codes, if you find a beautiful figure in the recently published paper and don't want to reproduce it or to reuse it for your own research, you can translate the figure into R codes and put your own data into it. Then you can also use Novi to polish your paper for publication. And we will go through the functions step by step. The first one will be the bioplot. We will show you how to make the plot as you like. For example, we can have the heat map. Sorry. So that let's take the heat map example, and we will show you the introduction and how the figure would look like, and the template data. If your data format is the same format, you can simply upload it and tell Novi to draw a heat map for me. And wait for 5 seconds, heat map will show up. You will see all the detailed information, x-axis, legend, and the other information. You can look at the heat map to fit your expectations, and all the other information also listed there, which you can, if you can download this figure for your presentation or publication later. And it's another example to work on the volcano plots. Again, just to upload your data, give the command, draw a volcano plot for me, wait for a second, the volcano plot will show up. And it also gives you all the information you needed. So all the color indications or the detailed parameters you can twist to make it perfect. And a single command sometimes might not be enough because you see that the Kinoplot's format is limited. But you can continue to give command to Novi, for example, to highlight the following genes, to change the font, to remove the background color, or to color based on a specific journal, or change the x-axis and y-axis, and to update the threshold. They can do all of them for you and show you the updated parameters you have here. Very handy to use. And you can also do it in the other aspect. For take this violin plot, for example, you have it. You can remove the sample if you don't like this low quality. You can apply y-axis transformation. Instead of log 10, you can make it log 2. And you can change the title, the image size, and the 7 different color to different groups. So in the end, it looks very professional here. And sometimes you might need more than one plot. You can ask the general bar plot. Then if you don't like this format, it's difficult to read the text here. You can switch the positions between the x-axis so that it's easier to read. You might want to also need to change the color. Okay, you can give the instructions back and forth. So that can maybe, if you don't like this red color, you can ask it to switch back to the older version. And sometimes you might want to merge different plots. So here I also created a bubble plot there for the same information. And for the bar plot, you can ask it to merge them together and for better presentation. And has also TechTalk modules. TechTalk answer your technical questions for your project. Also, you can use it for browsing, summarizing the latest publications in your research topic. For example, this one for the mouse transcriptomic sequencing, and summarizing all the recently published paper. You can ask it to put the papers in the table format, which journal come from, the one sentence summary, what it's doing. If you're using a different tool, you can ask Novi to put the papers in a different format, like a markdown table. And the another module is image to code, like I said earlier. If you find the recent publication has a nice figure, like this one, the circular heat map, which is cool. You want to use it for your own research, for your own project. You just gave this figure to Novi, and will give you back the R codes to generate this plot. And then you can apply to these codes to your own data. And to summarize, that all this request, the analysis can be supported in NovoMagic, so that redefine groups, remove samples, use a different tool, rerun enrichment, and adjust the figures, size, color, scale. And NovoMagic is limited to the downstream analysis, so that and the heavy step mapping to reads to the reference genome is not supported here. But all the downstream analysis from the quantification to the functional enrichment are supported. But all the other steps can be visualized. You can see what's happening there, but you cannot do reanalysis. But I think this is already very useful for the downstream analysis you can have. And the products available currently we support medical transcription sequencing and eukaryotic mRNA sequencing with reference genome. So, but in the end of this year, the Amplicon will be supported, and we also offer the other toolkits. And later, I will show you the demo reports of the live demo of this platform. Here, please follow me. If you're already a customer, you might already have the account of this customer service system. Just if you don't have it, you can ask a colleague that created one for you, not just in practice. for your future projects. Signing in, you will see how our platform looks like. Here, I wouldn't show you other details, but directly jump to the NovoMagic. Here you can already see some basic introductions, a few videos you can learn from. But I will show you on live here. So navigate to NovoMagic. And here, just the recent information, the news we gave you in the homepage. And here, the demo report you can request just to practice to use NovoMagic. And websites might be interesting for you. So you can simply click on the request demo, jump here, because the example I show here is already requested, you agree, but you can apply them here. Click on the user guide here, we show you we have all the videos to guide you step by step what to do. You can click on the products topic to show only the related videos for only human mouse or for other eukaryotic mammal-seq. And here our website and the older version of NovoMagic. Then here's also, maybe I briefly show you how the Novi looks like. Click on it, maybe take a few seconds to load. And you see the bioplot, TechTalk, image to codes, and paper polish, and how popular they are. Until now, it seems the bioplot, the greeting plot, is the most popular one. And clicking Get Started, you would enter the panel I just showed you earlier. But this NovoMagic platform itself is here. Click on My Project. You could see all the project you sequence with us. And click on Analysis, you would enter to the details. But if you click on the report, if you can see the report we already offered you, basically the same, and you can click through them, how they look like, and all the modules are there. Then coming back, you can have a look at all the results available here, and just the analysis you can do on this platform. And you can also read just to see which part you like or not like to plot. Yeah, just give us feedback and we further improve this NovoMagic platform. And I will get you to the analysis. And here you could see the name of the project and all the analysis modules I showed you earlier. Quality control, alignment, gene expression, differential expression, functional analysis, alternative splicing, SMP, fusion genes. But on this part, alternative splicing and Intel are not supported. Only the different gene expression and functional analysis are supported here. But in each of the module, you could see the top panel here. There's some information. If this is supported, you could see some information, but if it's not, you wouldn't see this. This project I show you is from Homo sapiens. It's just a test for the mRNA-seq data. It has already sample name, just fixed sample names. You can name them any way you like here again, and click update will change it to the new name, and you can group them any way you like. For example, I already just pretend that these are different concentrational drugs applied to cell lines, and you have different groups. Group 1, 2, 3, 4, you have them. If you find out later that, oh, I put this sample in the wrong group, I want to redo it, don't tell your supervisor, you can do it alone in this platform, do the rerun on analysis, give it a different name, for example, just test, and then to regroup all of them together. So like just T1 to include all the samples that have 9-1 in the name. To have 2 to have all the names 2 there. Yeah, just a example, you can regroup them easily. If you don't like the group you done earlier, you can do based on these older groups. For example, you find this one, actually this has a low quality, I want to remove it. You can click back and create a new group there. Or maybe put it in the sample into a different group. So put in another group, and it's all possible here. So it's really flexible to regroup your samples. And then you need to make the comparison scheme. And here you need to define your controls. If you're not sure, just to make everything compare with everything and do the filter out of the right comparison data. For example, using our just newly created test, there's no comparison yet. You can select comparing another test and to select the treatment group and the control group, add them and add another one. If you're supposing group 1 is a control, you can create a multiple comparison scheme. And after doing this, you could go down to the other analysis data. Here we just skim through this data. It's just summary and the data quality controls, sequencing error rate for all the samples. You can see them here again. And the GC content, sequencing data, filtering, and the statistical data. And there are also data for the mapping alignment and how the reads are distributed, how many the exons, introns, and the intergenic. Gene filtering doesn't matter. It's just the option for you to select, for example, the keywords. For example, if I'm working on immunology, I'm interested in enzymes, I can just type in enzymes and yeah, to show all the enzymes identified in this project that here get a list. Or you could search it based on other information. To give it a gene ID list and get all these gene sequences and annotations as you like. And here is a gene expression quantification, here where we start our real analysis. And there are some video tutorials you can look at, and there's nice introduction. This table itself includes all the reads you have. Here we have 58,000, which is quite a lot. If you already want to look into some of the data, you can click here, settings. And yeah, here you can decide whether to show the each column or not. And you can also decide whether to make any of the columns searchable. For example, if you want to search for gene description, coming back. For example, if you're interested in the kinases, you can just simply type kinase here so that all the genes that have kinase in the description would show up. See, like we have 10,000 in total. And you can create a gene site on them. Select all of them here and create a gene site, which you can use them later for enrichment analysis. But here I already have, if you already done it, you could see the gene site management here, which I did earlier, for the gene kinases, also enzymes. And then you can look at the gene distribution across the samples. Here are the analysis settings. You could rerun it. If you want to using different grouping scheme, you can select it. Or if you want to skip some samples, not to run them. For example, I don't like this E1. It looks so weird. I want to kick it out. You can unselect it and run the sample. Then you would get another box plot. You just briefly run it. See, after clicking it, it will be in this new task will be shown up here. If you click success, it show you all the box plot you created earlier. And sometimes it also showed up in the field if you've done something wrong, terrible. And the plot itself is interactive. If you can see here, if I put a mouse here, it will show you all the median, all the detailed information for each sample. If you click on any of the group here, it will remove them interactively. Here you can also save this new plot to the report. And you can also download the image here, like a different format we offer here, either vector or non-vector. The JPG and PNG or PDF and SVG are supported. And yeah, for each module, each function, we have the read button here. If you like it, you can let us know. If you don't, you hate it, you also gave just 1 star to make it, to let us know that we need to improve it. And then there's a correlation analysis. For each of the tasks I have finished, it will show the date being created, the detailed time, and also all the parameters. If you figure out anything's going wrong, you can come here to change it and then rerun it later. And there are these options. For example, if I want to include it or not, a whole group to remove some specific sample, and you can plot on group of samples to decide which algorithm to use and which logarithm option to use. And here, for example, it's the blue the color, the higher the correlation, but the red the color, the lower the correlation it is. You could notice these two samples are quite different, but these values are just 0.96, which are all right. If you find them extremely low, you can go back to the grouping scheme in the beginning just to make another group scheme based on this, just to kick these samples out of, like I showed you earlier. So it's really flexible, this workflow. And then you rerun the other analysis. So where were we? The correlation analysis. Yeah, again, you can save them to report and download the image. And the principal component analysis. Here you could see these two samples are indeed a bit different from the others, but they still belong to the same group. For each image I show you here, even for those that are not supposed to be reanalyzed for the quality control plots, they all can be adjusted by clicking the button here, image adjustment. So the global configuration, the width, the height, all the details. The theme configuration, give it title, to use a different color scheme. We can change the color for each specific one. For example, if we change the blue one to a green, we would immediately notice it. Or we click OK, and it will be updated here. And you can select a publication that the default color scheme and we applied it automatically. It's pretty easy to adjust. And all the other details, axis and legend. And yeah, here's just a brief introduction. So Venn diagram to show the genes expressed in either of the sample and it's shared. You can get the numbers of them, and down here you can have the gene list that expressed in all shared by all the samples. You can use them to create the gene set too, and yeah, these are all the information you can get from the quantification data. Then if you apply differential gene analysis here, you can check the default if you already in the report. If you found something's not done correctly, you can rerun it. For example, to use a different software at R or Lima. And if your sample contains batch effects, you can also remove them here. And the grouping schemes, you can choose a different scheme. Which groups to compare, either to use p-adjusted value or p-value, setting the threshold to determine significantly regulated genes, any other filters on the expression level itself, and yeah, to do the batch correction, which method to use, and which analysis. Here, then you can get after clicking the run later, you will get the output here, all the differential gene list. And here you can see the significance of each gene, all the comparison combinations for all the comparison there. And click further, you would see the differential gene statistics. You can give you an overview what happened in this comparison. All the significantly regulated, upregulated, and downregulated gene numbers. And then here you can see the volcano plots for each group of comparison. This figure can also be adjusted by clicking image adjustment, changing all the parameters you can imagine. Or later you can export this data to use Novi to fine-tune the figure as you like. For all the plots, you have the button to save the plot and download the image. Just a reminder. And then you have the Venn diagram to show how the different comparison, how many genes are shared. For example, this one has 248, it's shared by all the comparison, and lower part you would see the trees. We can make the compactor to have a better overview. We can either create a gene set on it, selecting them all, create a gene set, which I already did. But you can again see the gene set management here. Differential genes regulating all the samples, all the comparison, or you can download them and have a look in the Excel table. And cluster analysis to put all the differential regulated genes together to see what's happening among the samples. Clustering analysis, you can decide the clustering algorithm, either a group of samples, a mode, either to display genes or not, or choose color scheme. Sorry, very practical to use. And this, after deciding these differential regulated genes, we want to go to the functional analysis to see what's the real biological process happening behind. So here you can provide the gene set settings, so background gene set, If you don't provide, we use all the gene identified in this project. If you want to know the differential expression analysis, you can use the earlier decided the differential gene analysis defined earlier. So setting the criteria, choose the right corrections, and then click run. If you have a customized gene set, you can select it. For example, earlier they have selected kinases. You can do basically the same, or you only interested in the differential genes that are found in all the comparisons. Or you have a targeted gene list, you can decide them in your way. And after the enrichment, it gave you the histogram, the enrichment analysis. This is the latest one that we have. Yes, you can select between all these texts that have run earlier. So that the comparison group, which direction, or up or down. You can add all of them to the report if you like. And also the scatter plot is interactive. You can see all the information here and have up and down regular reports for the enrichment. And yeah, the idea is basically applies to all the other enrichment analysis. For the CAG, is also to select the differential analysis or use the target gene set, and then choose the p-value and adjust the p-value method. And then you, after running, you could look at the histogram and scatter plot. Yeah, the same applies to other regectome, DO, and the DisGeneNet. They're basically the same, or just switching between different databases. And you can run all of them together here using GSEA, same as before, differential expression analysis. To choose the right one and choose a comparison group and simply select all of them. So GO, CAG, reactome, DO, you can find all of them, and then click run here. Would get all the data here for all the plot you like. And the functional annotation of the oncogenes, so that to find out all the genes that are related to cancer. We don't support the reanalysis here, but just show you that you can look at it. The genes that relate to cancer or relate to disease. They are ready to be saved to the report or download. Protein-protein interaction network table to find out the relations between the identified and quantified genes. You can rerun them. Either customized gene set, differential regulate, express the genes, and run them. You would see this plot here. All these genes are interacting with each other. This information were from the based on the database from string. And for the rest of the modules, you can look at the data, but they're not supposed to be reanalyzed. For example, the alternative splicing. You can have all the list here, SAP and Intels, all these tables. And yeah, you can adjust the image as you like and then save it. So these are all the information I have for today for the live demo of the NovoMagic platform. If you have any questions, you can post them and my colleagues will help me to ask.
Hi, Wendy.
Hi, Carol.
Yeah. Thank you so much for the incredible and detailed talk. We do have many questions. They were popping right away while you were explaining. So I'll just select a few and see if we can answer them together. Okay?
Yeah.
And first we have Claudia, and she's asking if the analysis of the alternative splicing that we provide at Novogene, is it limited to annotated splicing isoforms, or can we actually detect intron retention or exon skipping events that are not found in databases?
I think alternative splicing, we can detect novel alternative splicing. Yes, only that you cannot redo it here. And the standard report we offer some novel alternative splicing.
Yeah. Okay, thank you. Now we have our next question from Dorothea, and she's asking if these services, NovoMagic, Novi, and so on, are they only accessible for sequencing data obtained by Novogene, or can they also upload the data from external services if the sequencing was done, for example, with the core facility?
Yes, for this NovoMagic platform itself, it only support the project you sequenced in Novogene in our company. But for the Novi here, you can save any data in the right format, not only sequencing data, any other data from your lab, from any other service, you can use our Novi. It's not a problem.
Okay, thank you. Now we have a next question from Vasilis. In the case of messenger RNA sequencing, what is the method used to filter out noise?
Sorry, could you please repeat?
Yes. So for messenger RNA sequencing, what is the method that we would use to filter out noise?
Filter noise. That's a quality control part that as I showed you earlier, that we have control the sequencing error rate, the DC content, and the contamination from the adapters. In the end, we would see only the clean reads here.
Okay, thank you. And we also got a question from Aisha. She's asking if there are any specific requirements regarding the format, the size, or naming of the files. Is there a sample or template of the file format that is available on NovoMagic so they can comply with our conditions?
Yes, for the samples, it's always suggested to follow the same pattern, to start with using only letters and numbers, and started with numbers. Better to not have space in between, use letters, numbers, maybe also underscores are supported. So the following standard, this makes it easier to avoid any error we would have. This is a really good question, and you should take care, could save a lot of trouble here. Thank you for this.
Yeah. So we usually have like this yellow coded text that would remind you of the rules, especially in the project analysis part. And if you're using Novi, for example, in the chat box right away when you're choosing the plot that you want to create, maybe volcano plot, maybe a bubble plot, Novi will provide you a demo example. So you can ask them. Like if you don't know what to do, you can also ask Novi, like, how should I proceed? And they will provide you a potential solution. All right. Now we got a question from Francisca. And they're asking if the changes made to a project using NovoMagic will be saved to the original project report, or is it possible to save the changes to a separate file project so that the original report remains unchanged?
The original report will stay the same. I can briefly show you here. For example, we make some changes, maybe. Yeah, we just rerun this group and we can go to the final report. You could see that the that the report is already updated. If you already click the button to save the report and go to analysis part, you could see all this plot here are updated based on what you have selected earlier.
Yeah.
So that you can download this report of just but also keeping the old one.
Yes. And in any cases, when you download the data from the CSS, which is the one that contains the FASTQ and the BIOM files, it always includes the original report too. So even if you accidentally make like a permanent change on NovoMagic, you will have the original report locally within the initial data release package. So we still have many questions to answer, but we have already reached our time of our webinar, which is 1 hour. We want to thank you again for all your questions. We will follow up with all the answers. It might take a few days. And if you have more questions, please feel free to keep writing here as I wrap up the webinar. And yeah, we'll reach out to you soon. So now for the further information part, if you have liked this webinar, just remind you that we have also diverse topics, not only from Novogene Europe, but also from other regions like the America region and also the EMEA region. Just feel free to scan the QR code here and you will get all this information. And for our upcoming webinar, we have Arman, our dear colleague here, who will talk about all-link proteomics. So if you're wanting to dive into this new field, feel free to register and we'll see you soon. Thank you so much and have a good day.
Thank you all.
Thank you.
Bye.